We finished a conversation on Tuesday. We talked about the balance, right? We finished hearing and talked about the balance, and today we're going to start and finish talking about smell and taste. So, anatomically, anatomically and physiologically, the function of smell is to warn about danger. Can be dangerous chemicals. For instance, if you feel the smell of gas, natural gas, and we'll talk about it as well, then you know that something is not going right, and you need to pay extra attention to that. Also, our ancestors, for them, sense of smell was a warning sign of predators or any other danger that can be posed by animals like lions and leopards and whatnot. Okay. Now, the basic idea, and it's kind of an arc in our conversation about physiology. If there is any sensation, do you hear me? Any sensation. We talk about interaction with the receptor, right? If it's a mechanical stimulus, it's going to be mechanoreceptor. If it's chemical stimulus, it's going to be chemoreceptor. If it's temperature, it's going to be thermoreceptor. But we may underappreciate how foundational it is. Any sort of interaction in the human organism is mediated by receptors. And in case of odors, in case of smells, the stimulus is always chemical, which means that those chemicals have to interact with their receptors, which are actual molecules, protein molecules, okay, and excite those chemical receptors. If you think for a second about chemicals being in the air, you can appreciate as well that the concentration of those chemicals in the air is very low. Does that make sense? It's low. However, once those chemicals end up on the surface of the nasal cavity, as soon as they are dissolved in the mucus that covers the interior surface of the nasal cavity, and they dissolved in a very thin layer of mucus. So thin layer, okay, you have a lot of molecules in that thin layer. The concentration immediately increases. That process, the situation, the fact that chemicals must be dissolved in the mucus to actually interact with the receptors is really important. If your nose is dry, you're not going to sense the smell as well. Okay? If you have too much nasal secretion, those chemicals that are around you are going to be really diluted, and the reaction with the receptors is not going to be well established too. So the amount of mucus should be just right. Does that make sense? Anatomically, the organ that actually senses the smell is olfactory epithelium, which is located in the roof of your nasal cavity. Now, I seriously doubt that what you see here is actual, up to scale image. It's very unlikely that the neurons that are responsible for the smell sensation are that huge. And they're just hanging from the top of your nose. Okay? So that olfactory epithelium is a pseudostratified layer of cells. Okay? And if you think about it, being positioned at the very top, the very roof of your nasal cavity, isn't the ideal position for sensing smell. Because when you are inhaling, the air usually bypasses the olfactory epithelium. Can you see that black arrow that I made? It bypasses the olfactory epithelium. Of course, there are going to be some chemicals that will reach and you will sense the smell, but think about this. Like, I'm here, even, if you pass the person, if you don't really sniffle, you often not gonna sense the smell of, I don't know, the perfume of that person, okay, unless there's a lot of it. So in order to increase the smell perception, we forcefully inhale by the nose, call it, we sniff, okay, and that forces the air up towards the olfactory epithelium. So far, does that make sense? The idea of position being not perfect. Structurally, olfactory epithelium consists of the supporting cells and sensory neurons. Okay? Does that make sense? Supporting cells and sensory neurons. Supporting cells support. They form that epithelium. They surround and sort of nurture the neurons, olfactory sensory neurons, that are in fact responsible for the sensation. On this picture, they're shown in yellow. You can see that at the distal ends of olfactory sensory neurons, right here, cells have cilia, so-called olfactory cilia. These olfactory cilia are covered by the mucus. Can you remind me, what is the function of mucus? What does it do? Dissolves the chemicals. Yes, exactly. It dissolves the chemicals. Does that make sense? So those cilia on olfactory sensory neurons, it's quite unique functionally. These cilia, unlike cilia on other cells, these cilia do not move. It is simply, covered by mucus, and essentially that olfactory cilia are functional dendrites. These olfactory cilia contain the chemical, the molecules, the receptor molecules, okay, that interact with the chemicals, airborne chemicals, once they are dissolved in the mucus. Chemicals interact with the receptors, they initiate the signaling. Is that understood? We're good? What produces mucus? Supporting cells and olfactory glands. Okay? Supporting cells and olfactory glands release the mucus. Mucus covers olfactory cilia, and when mucus dissolves the airborne chemicals, those chemicals interact with the receptors on the olfactory cilia. Let's write it down. Where receptors are. Okay? If you would, shine the light into somebody's nose, exactly at the roof, and look at the color of olfactory epithelium, it's yellow, just, little factoid. It contains the brown pigment and it gives it that yellowish hue. Olfactory neurons die, and they die pretty frequently. They are in sort of disadvantageous position. If you go into, if you are in the hot environment, if you are in the cold environment, if you inhale certain aggressive chemicals, I may have told you, maybe it wasn't you, I once cured my runny nose by inhaling chlorine. it was first year of college, we were making chlorine in the chemical in the inorganic chemistry lab. And properly, how you're supposed to smell aggressive chemicals like this. Well, nobody does it, trust me. When you have something in the tube, that's what you do. Okay, so that's what I did. It was indeed chlorine. My runny nose went away and my sense of smell went away for 3 days. Okay, because chlorine killed all the olfactory neurons that I had. Now, you can just, with any physiological, with any impact like that, you cannot just totally lose the sense of smell. So that is why there are so-called olfactory stem cells. Olfactory stem cells, they differentiate into the olfactory sensory neurons. That makes sense? So the complete turnover of olfactory sensory neurons happens every 1-2 days. 1-2 months, sorry. Does that make sense? So in a couple of months, you have totally new neurons in the nose. So far, does that make sense? So we figured out that in terms of the structural features, let's take a look at the olfactory neurons once again. These are what? Cilia. They serve as dendrites, right? Then you have soma, and then you have axons. Axons of olfactory neurons are non-myelinated, which makes perfect sense. Because smell sensation doesn't have to be that rapid. The axons of olfactory neurons go through the openings in the bony structure called cribriform plate, the ethmoid bone. If you would look at the anatomical image in the upper right corner, you will see the cribriform plate of the ethmoid bone. It is actually pierced. It does have those little openings. Bunches of axons here, okay, they form what is called filaments of olfactory nerves. So this bunch of axon and this bunch of axon, each of these are filaments of the olfactory nerve. Does that make sense so far? It's pure anatomy. Okay. Now that actually explains why sometimes there can be an infection, the meningeal infection and encephalitic infection, when you sniffle something pathological. Think about this. If any sort, if any sort of infectious agent manages to pull itself or push itself through these openings, where does it end up? It ends up in the olfactory bulb. That's where the filaments of the olfactory nerve go. And olfactory bulb is the first structure in the olfactory nerve. And if these guys are in the olfactory nerve, they're like a few steps away from the brain. Does that make sense? You may have heard about the condition called brain-eating amoeba. Have you? No? Well, some of you have. Brain-eating amoeba is amoebic disease which people acquire when they swim in warm waters, usually in the southern United States, when they snort the water, sometimes accidentally. Amoebic cells end up in the olfactory bulb. They pretty much penetrate through the cribriform plate, okay, go into the olfactory bulb, and then the person is pretty much doomed. It's almost untreatable, very hard to diagnose. People die in like 5-7 days. There were cases of laboratory acquired meningitis when laboratory personnel they opened the plate with growing microorganisms, accidentally inhaled a little bit of them, it immediately went into the through the cribriform plate in the brain, and that's it. Does that make sense? So that actually provides sort of a path from the nasal cavity to the brain. Does that make sense? Now let's talk about the sensations. I love this topic because for me, the mechanism of smell sensation is absolutely fascinating. Human sense of smell is decent. I'm not trying to compare ourselves to, say, dogs or cats or any other animal that has like tremendous sense of smell. But we can tell apart 10,000 different smells. Okay? Now, if you would look at the picture in the left lower corner of the slide, this one, you will see that many chemicals that smell completely different, many things that smell completely different, chemicals that are responsible for the smell are structurally similar. For instance, look at the vanilla and peppermint. They're somewhat similar, okay? You compare urine and amber, there are some similarities. You compare, I don't know, marigold and grapefruit, smells are different, okay? But chemicals are similar. I'm not saying they're the same, but they are similar. So, you see there are thousands of smells that we can feel. Each smell is represented by a certain chemical that dominates. How can we sense so many smells? The first answer that is most obvious is that we have a receptor. It's not there in the slide. I'm having conversation with you. The first most obvious answer is that we have a receptor for each smell. Okay? If there are 10,000 smells, how many receptors should we have? 10,000. Each receptor chemically, what type of molecule it is? I just told you 10 minutes ago. Is that like DNA or, I don't know, sugar or what? Receptor. Transmembrane. Receptors are transmembrane. Starts with P. Proteins. It's transmembrane protein, okay? Information about the protein is encoded where? In the DNA, in the form of a gene, right? So if you have 10,000 different receptors, you got to have 10,000 different genes. Does that make sense? How many genes does a human have? 20 to 30,000 total. So I would say it's very unlikely that half of the human genome will be dedicated entirely to just sense of smell. It seems like counterintuitive, and it's not this. Okay? So we can't have that many individual receptors. Linda Buck and that fucking guy, which name I don't remember. Okay? I don't remember. There was a guy who got Nobel Prize with Linda Buck. So two fellas identified the mechanism responsible for the smell sensation. They got Nobel Prize 2004. That's her Nobel Prize lecture. That's a picture from her Nobel Prize lecture. What they proposed, they suggested that each receptor can sense a certain number of odors. To understand this, let's take a glance at the table that I have on the right side of the slide. In the rows of the table are chemicals. In the columns are different receptors. Let's take a receptor number 19. You can see in the row how many different chemicals it can interact with. Can you see that? And also what this row represents, that it interacts fairly well with, say, hexanoic acid, but interacts very strongly with heptanoic and octanoic acids, and not so strong with nonanoic acid. So this receptor, based on the strength of interaction, can differentiate the subtleties in, between very similar chemicals. Do I make sense? So this receptor, if you would look at the table, can interact with 9 different chemicals. Does that make sense? So let's think about it one more time. Each receptor can interact with several chemicals. Is that understood? In the similar fashion, each order can stimulate a number of receptors. Look at bromo-octanoic acid. I just pick it completely just out of thin air. No pun intended. 8 receptors that bromo-octanoic acid can stimulate with different level of intensity. Does it make sense? Okay? Now, imagine, before we move on, imagine for a second that we try to smell certain pure compound, say bromo-octanoic acid. Bromo-octanoic acid will stimulate 8 different receptors, and they will be stimulated differently. Does that make sense? Think about this. If you play piano, imagine that you take a chord that consists of 8 different notes. Not only this, you play each note, or say you play a melody, okay? And you play each note, some of them are louder, some of them are quieter. It's a very distinctive melody. Does that make sense? Do you understand what I'm trying to say? Now imagine you are, look, come on. How many of you smelled bromo-octanoic acid? I mean, I did a year of organic chemistry lab. I never smelled bromo-octanoic acid. So unlikely that any of you did. It's not like very common thing in the household. Did you smell cinnamon? Yeah. Cinnamon smell is probably not, do I have cinnamon? No, I don't have a formula, but there isn't like aromatic acid that's responsible for that cinnamon smell, but probably it's not the only one chemical. Say it's 2 or 3 different molecules that dominate in the cinnamon smell sensation. Do you follow me? Each molecule will interact with several receptors. And these 3 molecules will produce like a symphony. Does that make sense? And that symphony will be unique based on the concentration of each chemical and, the nature of it. Does that make sense? Turns out we can get away with 400 smell receptors. This is it. Okay? So 400 smell receptors enable humans to distinguish 10,000 different odors. Now, another thing that is important. Imagine you have, imagine a dendrite. Okay, you have a dendrite. That's a dendrite. Okay? That blue line is the membrane, right? Where are the receptors? Here. Does that make sense? Receptors are in the membrane of the dendrite. Do you follow me? Now, if those receptors can sense different odors, if those receptors are different, what's going to happen? It's going to be very confusing because the same neuron will send a huge multitude of signals. And what it comes down to, think about this. What it comes down to, the signaling in any case, it doesn't come down to the receptor. It comes down to the transmission of electric signal from the neuron. So neuron must send unambiguous signal that can be interpreted in a very specific way, which essentially means that each neuron, olfactory neuron, has only one type of receptor. Let's write it down. All receptors are of the same type. Does that make sense to you? If you want an analogy, and I don't know if you do or don't, but I'm going to provide it to you anyway. Think about cones. What three types of cones do you have? Red, blue, and green. Red cone has a certain spectrum of sensitivity. Blue has a certain spectrum of sensitivity. Does that make sense? Now imagine that same cone has photopigments that can sense red or blue or green. So same cell will be sending signals corresponding to all parts of the spectrum. Will be able to distinguish the colors? No, you will have like white light for everything. You will actually see the white light. So same goes for these guys, for the olfactory neurons. They just have more modalities. Not just red, blue, and green, but there are 400 types of receptors. Does that make sense? Now, again, if you sense a particular smell, you will stimulate, say, 8 different receptors. Those 8 different receptors will be located on 8 different kinds of neurons. So you will stimulate 8 different kinds of neurons. And they will have a unique combination of signals. Does that make sense? This is called combinatorial receptor codes. Okay? And it is pretty nicely exemplified on this picture. Okay? Here you have odorants and you have receptors. I took this picture and modified it slightly from that Cell paper published by Linda Buck. Okay? So if this is the odorant, it will stimulate this receptor and this receptor. Does that make sense? The green portion stimulate this green, the red portion will stimulate this red. If it's such slightly different combination of chemical parts, this violet one will stimulate the violet receptor, the red one will stimulate the red receptor. Of course, there are no colors, but the point, I hope you can see the point. It's a combination, chemical combination of parts. Does that make sense? Okay. And another sort of on the side notion, how long does it usually take for a person to get a Nobel Prize? Actually, in like mid-20th century, it could be 30, 40 years before between the discovery and the Nobel Prize. That's the paper, 1999, like the most important paper describing the sense of smell, the mechanism of smell sensation. Of course, it wasn't the one. They were like working on it for probably a couple decades. But it was like the most important paper. They get Nobel Prize in 2004, 5 years later. Now, Nobel Prizes, they given like, okay, that's important, let's give it Nobel Prize. Too many people died before they could get Nobel Prize. Do you understand that mechanism? So, single chemical may stimulate many receptors. One receptor can be stimulated by multiple chemicals. Every neuron has only one type of receptor. We clear? Good. Can we move on? We're moving on to the fun part. The molecular mechanism of the smell sensation. We again looking at the second messenger activation. This blue here is mucus. Odorant, the chemical, is dissolved in mucus, okay? And when odorant binds to the receptor, it activates G protein. Again, it's a G protein, okay? Actually, these receptors that can activate G protein are called G protein coupled receptors. Do you follow? G protein is activated. It moves, translocates, and activates the enzyme called adenylate cyclase. The function of adenylate cyclase enzyme is to convert ATP into the molecule of cyclic adenosine monophosphate, or CAMP. Before we move any further, I want to say that this part that I highlighted here is the common part of various signaling mechanisms, including signaling in endocrine system. If you know this mechanism now, it will be much easier for you to study endocrine system later. Do I make sense? I hope it serves as the good incentive for you. Cyclic AMP is the very powerful signaling molecule inside of the cell. And in this case, cyclic AMP binds to the sodium-calcium channel in the membrane of the olfactory receptor. Does it make sense? When CMP binds to this channel, it opens it. What type of channel that is? What type of gating? Is that mechanically gated, voltage gated? What do we call this? Channels that are opened via binding of chemical. Say it? Ligand gated, right? Ligand gated channel. So that's the ligand gated cation channel. CMP is the ligand, and it opens the channel. Sodium and calcium flow into the cell. As sodium and calcium flow into the cell, what happens to the membrane potential of the cell? It goes more positive, and we call this, say again? Depolarization, yes. So olfactory neurons depolarize. That creates graded potential on the olfactory neuron. Do you follow me? And olfactory neurons release neurotransmitter. Okay? That makes sense? Okay, they release neurotransmitter. Neurotransmitter that olfactory neurons release acts on so-called mitral cells. So essentially, olfactory neurons are the first order neurons. Mitral cells are the second order neurons. The synapsing between olfactory neurons and mitral cells occurs in the structures called glomeruli. Okay? Glomeruli and mitral cells are located in the olfactory bulb. That is on top of the cribriform plate of the ethmoid bone. Actually, I have a skull model right here, and I'm going to walk around and show it to you. So if you would look over here, see those pretended holes, perforations? This is where the olfactory bulbs are located. That's the cribriform plate of the ethmoid bone, just top of it. Okay, see that? Right here. So those perforations, that's where, the filaments stick out. So that should be, the olfactory bulbs should be here. I mean, they're just gone, but, okay? Does that make sense? So here you have synapsing. Now, that's another important thing, is that how many different types of olfactory receptors a single olfactory neuron has? One neuron? Huh? How many types? One neuron? One type, right? All neurons with the same type, they converge on the same glomerulus, which makes perfect sense. Does that make sense, right? Think about this. You have many neurons that perceive, the same chemical, have the same type of receptor. They're all going to be stimulated together by the same chemical. They all will, if there is a chemical, all those neurons will generate graded potential. And as a result of graded potential, neurotransmitter will be released into the synaptic clefts between the olfactory neurons and the mitral cell. And all those inputs, all those graded potential from each olfactory neuron will summate on the mitral cell. What type of summation that is? When all those neurons at the same time will release neurotransmitter, and neurotransmitter will generate action potential in the mitral cell. What type of summation that is? Temporal or spatial? Oh wait, spatial. Because at the same time, in different locations, right? Does that make sense? So that's a spatial summation. Example of spatial summation here. Now, if you think about it, you have an order, which, for the simplicity, let's assume you have an order that's represented by one chemical. Do you follow me? One chemical, how many receptors can it stimulate? Hint, I don't look for the number. How many receptors? One or more than one? More than one. So there are going to be several sets of neurons stimulated by a chemical. Does that make sense? The sets of neurons will synapse. Each set will synapse at the unique glomerulus on the unique mitral cell. So each order will stimulate a unique set of mitral cells. Again, analogy of the playing chord. Okay? Certain set of mitral cells stimulated will correspond to a particular order. Mitral cells refine, amplify, and relay the signal. Now, very important thing. They have high threshold for activation. Right now, okay, let me put it this way. Are you overwhelmed with the smell sensations on the regular basis? Like you're sitting at home, are you like really like crazy, like overwhelmed by all the smell? No. Let's be honest. I mean, obviously, you sit at the dining table, you can smell the food and stuff like that, but it's not like you have enormous sensory input from your nose. Do I make sense? This is due to the high threshold of activation of mitral cells. In fact, mitral cells are sort of constantly inhibited by amacrine granule cells. It prevents us from being overwhelmed by the smell sensation. Now, if you would compare the threshold of activation in humans and in dogs, which species have higher threshold of activation, humans or dogs? Humans, of course. Dogs have lower threshold of activation, so they can sense way more smells. Does that make sense? And I want to highlight, often you can hear the words, such and such animal evolved to do that. Nobody evolved to do anything. We don't have a very good sense of smell because we didn't need it for survival. If we would need it for survival, we would have it. So those, like in dogs, why they have great sense of smell? Those dogs, and wolves or whatnot, they didn't have a great sense of smell, they died. Okay? If you don't see a particular type of a trait, carriers of that trait died years ago because they just didn't survive. They were not unfit. Does it make sense? Now, transmission of the signal. Receptors to the olfactory bulb and through the nerve to the various structures in the brain. Okay, the piriform cortex in the insula, olfactory tubercle, amygdala. What I want to highlight is that there are two major parts of the brain that participate in the smell response. One, frontal lobe and thalamus. That's conscious interpretation of smell. Did it happen to you when you sense the smell and it brings back memories? It's actually a very strong response in humans. For me, it was sort of funny. The first place in the U.S. where we lived was Nebraska, which is like, you cannot find anything more Midwest. And when we arrived, Nebraska had a very specific sense of smell. It was pleasant, but it was just very, very special smell in the air. We lived there for a year and then moved to Louisiana. And the moment we step out of the car, Louisiana smells completely different. We lived there for 8 years. We moved back to Ohio. So my wife and I, we visit Cleveland to look for a place to live. And we step out of the airport. It's in the summer. And Ohio smells just like Nebraska. So it's Midwest. And I remember that smell. I mean, I wouldn't be able to describe it. But I remember the smell. And you remember smells too. So that's your frontal cortex. Okay? That's how you consciously perceive smell. Another important part is hypothalamus and amygdala. Okay, limbic system, emotional responses to odor. Hypothalamus would be, for instance, responsible for tasty smell sensation. Okay? Dangerous smells will activate sympathetic nervous system. It isn't so, obvious in humans, but you know how there's an expression that if you are afraid of the dog, even if you don't show it, they can smell it. There is some scientific basis under it. When you're afraid, you produce certain chemicals. You are producing chemicals when you are scared. Dogs can sense those chemicals, and perhaps that's how they know you are scared of them. That makes sense, right? Protective responses. If the smell is extremely unpleasant, it may trigger things like sneezing or choking or vomiting. Did we talk about ethyl mercaptan story? The guy that synthesized ethyl mercaptan. We didn't? I love this story. Absolutely. I guarantee, I'm not kidding. It's a true story that happened in my alma mater, School of Chemistry of Moscow State University. So ethyl mercaptan, you don't have to write the chemical formula, but it's this. Yes, is considered to be the most disgusting, odor in the world. It smells like shit, but very strong, very strong. You really need very tiny concentration to sense the smell. And it is fairly easy to make in the lab. You can do it. It means organic lab. You can do it. There's one guy, third year, organic chemistry lab, and he decided, why don't I make it? He makes it, and it's in the little tube, and it's very volatile. It's pretty much a gas. You need to cool it down to make it a liquid. It's a gas. It's volatile, and the tube is closed with that rubber stopper. Well, this rubber stopper isn't a barrier for ethyl mercaptan, and ethyl mercaptan starts to sort of, leak from the tube in the surrounding air. And since the concentration that you need to sense the smell is very low, people on the other side of the lab start to question, who shitted in the lab, because it smells like somebody just did. The guy understands that he's going to get busted. So he runs into the hallway. Well, not runs, I need to go to the restroom. So he walks, hiding the tube. And he needs to dispose of it. And he understands he cannot just dump it in the lab, because that would be like everybody would start just choking and whatnot. So he walks in the hallway, and there's a toilet. I know there's a toilet next to that lab. So he walks in the hallway, sees the toilet. The smell is almost intolerable. So he opens the door, throws the tube inside, and closes the door. He can't really carry it. He understands there's no way to do that. Obviously, the tube is glass, so it breaks down. He doesn't see it. He comes back to the lab. Everybody's happy. The hoods, ventilation system pulls the smell out, so it's fine. A couple minutes later, some unlucky dude decides to use the restroom. Okay? So I want to remind you the tube is broken. So everything, all the content is in the pretty much air. He opens the door. And the moment he opens the door, he vomits right away. I mean, it's physiological response. He doesn't even think about it. He doesn't even try to hold it. There's no way you can hold it. It's so physiological. Okay? So that is a response to unpleasant smell. There was another great story. Actually, all sulfur organic compounds that have sulfur, they smell very badly. People who work with those compounds, if they walk into the subway hobos leave. They cannot stand the smell. So, I mean, you cannot like wash your hairs to get rid of the smell. You have to shave. You throw away your clothing because you can't wash it. It's just horrible. So there were two chemists at the University of Harvard. I think it was Harvard University that worked with pseudoorganic compounds, and they were asked to leave the campus. University was willing to provide them some place to work, but they said it's impossible. The smell that you generate in the lab is intolerable. So they found a barn at the pig farm. And the owner of the pig farm said, yeah, sure, I'm used to all the smells. So they installed the lab in that barn and started to work. And a couple of months later, the owner of the pig farm told them, can you please leave? The smells that you produce in the lab are intolerable for me. Okay? It kind of gives you a perception how bad the smell can be. Pseudoorganics are horrible. Some people experience anosmia. It's an ability to sense the smell. Parkinson's disease, the deterioration of the neurons, can cause anosmia. Head injury, inflammation of nasal cavity, they all can cause it. Okay, very common cause of anosmia is the blow into the head. When cribriform plate is broken, the filaments of the olfactory nerve are damaged, and there's no transmission from the olfactory neurons to the mitral cells in the olfactory bulb. Uncinate fits is the fancy name for olfactory hallucinations. Usually they are present as the unpleasant smell. Olfactory auras are transient uncinate fits, transient olfactory hallucinations. It's a very common symptom of the oncoming epileptic bout. People with epilepsy often experience uncinate fits immediately before the epileptic bout. Does that make sense? Any questions? Say again? It doesn't cause, it's a symptom. So for instance, I have a student in one of my classes, and she very infrequently, she has a seizure. It never happened to her in the class, but she told me, look, I know when it comes because of this olfactory auras. Pretty much she tells me, I will tell you, I'm about to have a seizure. Because, you immediately start to sense this smells. Okay? So the last sense that we're going to talk about is taste. The anatomical parts that are responsible for taste sensation are called taste buds. Mostly in the tongue. Some of them are located on the soft palate. Obviously, you can't see soft palate on the cranial model, but it's about here. Cheeks, epiglottis, and pharynx, all the way down the throat. Okay. Taste buds are located in three different types of papillae. The valate papillae, the large, roundish valate, fungiform papillae that are located in the anterior two-thirds of the tongue. Fungiform quite literally means shaped like mushrooms. And foliate papillae. Folia means leaves or folds. You can see those little folds on the side of the tongue. Okay. Now this picture shows you a representation of the taste bud. Each taste bud consists of multiple gustatory epithelial cells. On this picture, they're shown in that, from your vantage point, it's probably going to be violet. Okay, those are gustatory epithelial cells that are responsible for the taste perception, and so-called basal epithelial cells. On the apical surface of gustatory epithelial cells, are gustatory hairs. You can see the gustatory hair exemplified here. Gustatory hairs are microvilli that extend in so-called gustatory pore. These microvilli are surrounded by saliva. Does that make sense? And in fact, saliva is absolutely critical for your taste sensation. Saliva dissolves food chemicals. If you have no salivary production, and some people have this problem, if the person has head and neck cancer and the part of the head or neck has to be surgically removed, which happens, and the person loses some of the salivary glands, the amount of saliva will be greatly reduced. And that reduced amount of saliva will, one of the consequences of this will be an impaired taste sensation. So far, does that make sense? So, those gustatory hair cells have receptors on the hairs. Hairs are the structures responsible for the sensation of taste. And you can see that dendrites of the nerves that transmit taste sensation, like facial nerve or glossopharyngeal nerve or vagus nerve, those dendrites surround the basal aspect of the gustatory cells. Okay? Gustatory cells can release two major types of neurotransmitter, serotonin or ATP. We'll talk about it. We'll talk about, release of ATP as a neurotransmitter. So far, do you follow me? Finally, think your gustatory epithelial cells are even at the higher risk of being killed than your olfactory neurons. If you drink something hot or eat something spicy, those gustatory cells will be killed, will die. So they are replaced every week or week and a half via differentiation of so-called basal epithelial cells. In this image, on the right side of the slide, basal epithelial cells shown in blue. So please understand the difference between basal and gustatory epithelial cells. Know which papillae contain taste buds. Understand what the gustatory hairs are. And understand that saliva is necessary to dissolve food chemicals for the proper taste sensation. We cool? Types of taste. What I want you to understand, not only know all 5 types, I want you to understand which chemicals can stimulate each taste type and what is physiological importance of taste. For example, sweet, obviously sugar, also various types of alcohol, lead paint. I'm really curious who tried it, but what is the physiological importance of the sweet taste? Why do we need to taste? Why do we have to have an ability to taste something sweet? Why do you want to know that this is sugar, that this is sweet? Sweet foods, what are they rich in? Sugar. Why do we need sugar? There you go. So that allows us to taste energy-rich foods. Does that make sense? Sour. Sour taste is sensation of hydrogen ions, essentially sensation of acid. Why do we need sour taste? What is sour? Which food is sour? Huh? Yes. I mean, but physiologically, well, lemons, I don't really see people around like just crunching on lemons. Usually it's spoiled food. Does that make sense? We need to know when the food is spoiled. When it tastes sour, it's spoiled. Of course, we started to trick. Nobody will call sauerkraut spoiled food. Okay, lemons are not spoiled. Lemons are sort of exception, but generally, spoilage of the food. Salty. Salty food, well, salty taste is mediated by the ions of metals, mostly sodium. What is the physiological importance of tasting salty foods? Why do we need sodium chloride? What? Fluid. You're right, fluid balance, more generally. Electrolytes. We get electrolytes, right? Okay, make sense? We good? So that's electrolytes. Bitter. Bitter taste in our day-to-day experience, quinine, nicotine, caffeine, morphine, strychnine. I mean, some of those chemicals are pretty good, like caffeine, good, right? It's your coffee. Quinine, very bitter. It's a drug against malaria. Nicotine is in the cigarettes, but we don't like drink nicotine. Morphine is opioid drug. Strychnine is the poison. Generally, bitter taste gives us a clue. If something is bitter, it's probably poisonous. Does that make sense? Alkaloids that you can see here, that word alkaloids, natural alkaloids are often poisonous to humans. When we taste something bitter, we probably shouldn't eat it. Make sense? And finally, the fifth taste is umami. Umami is delicious taste. Amino acids like glutamate and aspartate. Where can you find umami taste? What is that delicious? I'm trying to think of the... Okay, I remember the English word. Savory. Which type of food will be savory? Huh? Steak, huh? Well, I mean, last time I checked, there's no gravy running around, but there are steaks running around in the forest. I mean, they're unprepared, right? Well, think about evolution. Think about us like a million years ago, right? So, chicken, any type of meat or poultry, that's umami. Cheese is umami. Milk, any... Now, what's common between cheese, milk, meat is not dairy, like chicken, beef, pork, they are rich in what? Protein-rich food. Umami gives you an idea this food is rich in protein. Does that make sense? I would never call like celery savory because it's not rich in protein. Does that make sense? So these are 5 modalities. Please know them 5 and know what stimulates each modality and what's the physiological importance of each. Okay? There's some evidence that we can taste long-chain fatty acids, which says fat oils. Okay? Now, here's the important thing. Taste buds usually have a mixture of the taste receptors. So taste buds respond to all modalities of taste. The idea that, like, sweet sensation is, I don't know, on the tip of the tongue, no. every area of the tongue, as demonstrated, can sense any type of taste. Does it make sense? Okay? Any area of the tongue can sense any type of taste. But of course, every receptor is responsible for a single modality. Receptor for sweet will sense sweet. Are we clear on that? Now the physiological portion, the molecular portion. How the chemicals, taste chemicals, sometimes they are called tastants. How those taste chemicals can be transduced into the nervous signal. The simplest one is salty. If you eat something salty, salt is sodium chloride, right? Do follow me? It's sodium chloride. So when you eat something salty, concentration of sodium in your mouth is high. Concentration of sodium inside of the cell is low. Sodium simply diffuses through the sodium channels into the cell. Is that clear? As sodium diffuses into the cell, what happens to the membrane potential of the cell? Cell depolarizes, releases neurotransmitter. Are we clear? Is that simple enough? I think that's pretty simple. Sour taste. There are a few mechanisms. One is sodium channels that are gated by protons. Does that make sense? In the presence of hydrogen ions, sodium channels open, sodium flows into the cell. Another mechanism is direct influx of hydrogen into the cell. Hydrogen ion, how is it charged? Huh? Hydrogen ion, how is it positive? As it flows into the cell, what happens to the membrane potential of the cell? Cell depolarizes again. Does that make sense? Okay? Now, bitter, sweet, and umami. So we're going to focus on these ones. Bitter, sweet, and umami tastes. They transduce the sensation by a second messenger system. Okay? So one mechanism, for instance, for sugars is activation of adenylate cyclase. If we will go back for a second, what does adenylate cyclase do in general? Produces cyclic AMP. Does that make sense? Sorry. Now, cyclic AMP in this case works as the ligand, but instead of opening channels, it closes potassium channels. Concentration of potassium is higher on which side of the membrane? Is it higher inside or outside of the cell? Potassium. Inside. So potassium generally diffuses out of the cell or into the cell? Out of the cell. If you close potassium channels, is it going to diffuse? No. Potassium, how is it charged? Positively. So if positive charges are not diffusing out of the cell anymore. What's going to happen to the membrane potential of the cell? Depolarize, because you have positively charged potassium ions accumulating inside of the cell. Do I make sense? So that's the explanation why it's depolarized. Okay? Some sweeteners and alkaloids that mediate bitter taste. They will activate the enzyme called phospholipase C. Phospholipase C will produce the chemical called IP3. Let me go forward for a second. This shows better, okay? So you see you have receptor, taste GPCR. It activates phospholipase C, and phospholipase C produces IP3. So far, do you follow me? You don't? Okay. Give me a sec. So what I want to do then, I want to draw, because you've encountered adenylate cyclase before. You know what I'm talking about in this case, right? But we didn't talk about phospholipase C yet, so we will try to draw it. Sounds good? Okay, let's just, kind of remember phospholipase C. Think about another thing. Umami and bitter taste. When you have channels, potassium channels that are gated by GMP, cGMP, if you destroy cGMP, what's going to happen to those channels? You have channels, potassium channels, that are open by cGMP. If you destroy cGMP, what's going to happen to those channels? They're going to close. If potassium channels close, what happens to the membrane potential of the cell? Depolarizes, because potassium stays in the cell. Now let's talk about phospholipase C, all right? This is membrane, okay? This is the receptor. This is the tastant. Are you with me, James? G protein is bound to the cytoplasmic, so it's cytoplasm. Cytoplasmic end of the receptor. When the tastant binds to the receptor, G protein gets activated. It translocates to the enzyme called phospholipase C, PLC. Okay? Does that make sense? Phospholipase C. Phospholipase C produces the chemical called IP3, phosphatidyl inositol 3 phosphate, if you're too curious to know the full name. Okay? Wait, that's the right name. Phosphatidylinositol 3, okay? IP3. The function of IP3 is to open calcium channels that are in the endoplasmic reticulum. So this is endoplasmic reticulum. This is the calcium channel. IP3 binds to that channel, opens it, and calcium is released into the cytoplasm. Does that make sense now? If you would look at this image right here, that's what it tells you. Binding of tastant, let me erase it completely. Binding of tastant to the receptor activates G protein. G protein activates phospholipase C. Phospholipase C produces IP3. IP3 opens the calcium channel. Calcium flows out into the cell. This calcium causes two things. First, it opens sodium channels. Sodium flows into the cell. Influx of sodium leads to which change in the membrane potential? Depolarization. And also, it opens ATP channels. ATP is released from the cell, and in this case, ATP works as the neurotransmitter itself. Does that make sense? Okay? Okay? To get it. Now, I want to quickly review with you this idea. Conversion of cGMP to GMP and closure of potassium channels. This is the receptor, okay? This is the enzyme called phosphodiesterase. And this is the potassium channel. Normally, potassium, that's the cytoplasm. Normally, potassium, where does it diffuse? Which direction? Out of the cell. Okay? This is G protein. Let's see what's going on when umami or bitter tastant bind to the receptor. As they bind to the receptor, what happens to the G protein? Again, you work in solo. Help someone, help Alexis. So G protein gets activated. What happens to the G protein then? It moves where? To the enzyme. What does it do to the enzyme? It activates it. What does this enzyme do, phosphodiesterase? What does it do to cGMP? What does it do? Destroys it. It degrades cGMP. So cGMP is degraded to GMP. cGMP keeps the potassium channel open. As the cGMP is degraded, what happens to the potassium channel? Say again? It gets closed. Is there any efflux of potassium now? No. And potassium, as it is kept inside of the cell, what happens to the membrane potential of the cell? More positive, meaning depolarization. Does it make sense? Now, what I truly want you to know, I'm not going to ask you about the exact matching, like sweeteners stimulate phospholipase C, while umami stimulates phosphodiesterase. Okay? Or sugars stimulate adenylate cyclase. No, don't worry about this. But I do want you to know the mechanisms behind adenylate cyclase, phospholipase C, and phosphodiesterase mediated second messenger systems. So if I tell you the function, umami taste stimulates phosphodiesterase, which breaks down cGMP and closes potassium channels. You have to know that, okay? Does that make sense? But I will not ask you which taste stimulates phosphodiesterase. Umami, bitter, sweet, or salty. Don't worry about that. Am I clear? You have to know the mechanism for salty, influx of sodium, and mechanism for sour, interaction, either opening of proton-gated sodium channels or direct influx of hydrogen ions. Does that make sense? Obviously, different receptors will have different sensitivity, which means there will be different activation thresholds for different tastes. The bitter taste is the most sensitive, since it's protective, naturally protective. This flow chart here gives you a perception of what's going on. When cell experiences the influx of sodium or accumulation of potassium, cell depolarizes, neurotransmitter is released, and that neurotransmitter will generate a generator potential in the sensory dendrites. And, fibers, nerve fibers will transmit that signal to the brain. There are 3 nerves that carry taste sensations from your mouth. Facial carries from the anterior 2/3 of the nerve, or of the tongue. Glossopharyngeal, posterior third, and vagus nerve carries taste sensations from the epiglottis. Okay? Fibers synapse to the second order neurons in the solitary nucleus. Second order neurons extend all the way to the thalamus, where signal gets relayed via the third order neurons to the cortex in the insula and into the limbic system. Because you have those responses, right? When you taste something very, very unpleasant to you, what is your usual response? Well, facial expression may change, but vomiting. Okay, if you taste something really unpleasant, it may cause the vomiting reaction, the nauseating reaction. Does that make sense? That's your limbic system response. And come on, I don't know, some people like chocolate, some people like steak. Okay, so your just sense of happiness will be brought by different tastes. That's it. That's your limbic system response. Now, anomalies of the taste sensation are not common because of the 3 nerves. Chances to damage all 3 at the same time are very low. Okay? What can affect the taste sensation? Well, various types of head injuries that will affect the nerves, chemicals, radiation treatments, anything that can affect the nerve. There is another thing. Your taste sensation is very important, is supported by your smell sensation. When you have runny nose and you eat, food tastes differently. Why? Because you do not perceive the smell. It was actually sort of demonstrated. It was a funny study, I suppose. People were given coffee and they had like open cup and the cup that completely isolated their nose from the coffee aroma. Turns out people enjoyed the closed cups much less than the open ones. When you deprive a person from the smell modality, it's not as good. Does that make sense? On the other hand, have you ever heard about the food called lutefisk? It's the Icelandic food, which, as far as I remember, cod, the fish, which was fermented for a very extended period of time. It's prohibited in public places in Iceland because of the smell. I read an account, the person tried it. So pretty much the conclusion, you can eat it. The only condition, you have to close your nose. I mean, if you have both modalities, you're going to vomit. It's just inevitable. It's so strong, the smell and the taste together. If you deprive yourself from the smell, then, you can eat and swallow. Okay? So any type of nasal congestion will impair your taste. Questions? Before we take a break, I want to ask you, was I explicit enough about this, the mechanisms? I'm not going to ask you, umami taste stimulates blah, 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 blah. You have to understand, umami, bitter, sweet, second messenger. Salty, ion channels. Hydrogen, sour, ion channels. Are we clear? Okay, let's take a break.